So do we have any evidence for this? Well, if we take keratinocytes from a mouse and we heat up those keratinocytes to a painful temperature, or we don't, and then we collect the medium that was surrounding those cells, let it cool down to room temperature, and then we splash that onto sensory neurons in a dish, what we find is that indeed there are sensory neurons that will respond to the heat-treated medium, but not to the medium from keratinocytes that were grown at room temperature. Okay. So what are some of the signals that are involved? Well, here one example of a molecule that's released by keratinocytes when you heat them is this molecule called prostaglandin E2. I'm not going to go into great detail here, but just say that it's a lipid molecule that's involved in pain and inflammation, and it is released from keratinocytes when you heat them in a temperature-dependent fashion. And what we found is that if we make a mouse where we have increased the level of expression of one of these heat-gated TRP channels, TRPV3, within the keratinocytes, that those keratinocytes release more of this prostaglandin E2. Now, if we go in the other direction, if we take a mouse that is missing TRPV3, in this case, we find that we get some deficits in heat sensation. So this is a little bit of a complicated one, but I'll just say that it's very, that if you put a mouse in a thermal gradient, so you give it an opportunity to choose between a very cold floor temperature or a very hot temperature or everything in between, mice pick their preferred temperature range, just like people do. And on some genetic backgrounds, it doesn't matter whether or not those mice are lacking TRPV3. But on other genetic backgrounds, it turns out that whereas wild-type mice show a fairly broad distribution, where they'll choose anything between 20 and 35 degrees Celsius, these TRPV3 knockout mice tend to prefer cooler temperatures. Okay? They're less likely to avoid the warmer temperatures. And so this tells us two things. First of all, it says that TRPV3 is indeed involved in the perception of temperature, but it also tells us that just like in humans, genetics matter. In other words, the genetic background of the individual can strongly influence what sensory pathways are engaged. Now, if we're talking about painful heat, there also seems to be a role in these heat-gated channels. So, for example, if you have mice that are lacking both TRPV3 and TRPV4, they show a slightly delayed response to heat in that tail immersion assay I showed you earlier. But you'll notice that's only a partial effect. It's a very subtle effect. And we think that's at least in part because keratinocytes have other mechanisms to detect temperature besides TRPV3 and TRPV4. In fact, if we go back to the measurement of the prostaglandin release in response to heat, we find that there's only a very small deficit in the absence of TRPV3 and TRPV4. So it tells us there must be other mechanisms for sensing temperature in these cells. So what are the questions that are occupying us and trying to understand this system? So first of all, we'd like to know, what are the non-TRPV3, non-TRPV4 sensors for heat in keratinocytes? Another thing that now we can answer using some more powerful transgenic mouse tools is to ask the simple question, if you only stimulate a keratinocyte, is that sufficient to cause a perception of pain? And if so, what are the chemical messengers that mediate this communication between keratinocytes and sensory neurons? And which of the particular neurons are actually doing the listening? Which of the many subtypes of nociceptive neurons are responsible for receiving that information? And then finally, do other non-neuronal cells of the skin, besides keratinocytes, immune cells, glial cells, and so forth, do they also act as virtual antennae for the nervous system? Why does this matter? Well, there are a number of reasons why this might be worth understanding. First of all, a better understanding of the causes and contributors to chronic pain might emerge, and we might also understand better why certain skin diseases and inflammatory diseases promote chronic pain. Secondly, the fact that these events are going on way out in the periphery might allow us to devise better therapeutics for pain that avoid the central nervous system side effects that you might get from, for example, from opioids. You know, one of the problems with using morphine and other pain-killing opioids is that in some cases you can get CNS side effects such as respiratory depression or addiction. So if you could develop therapies that target those peripheral events, you might be able to sidestep some of those side effects. And more broadly, because you might be able to deliver this therapy topically in a very local area, you can achieve concentrations that are necessary to reduce pain, but not have other systemic effects like GI bleeding that you get, for example, from taking lots of aspirin. Okay. And then finally, I just want to mention real quickly and amplify the point that was raised earlier, is that pain research at Hopkins is really entering an exciting phase because we have a number of of entities now that some of them self-assembled, some of them assembled as a result of administrative entities that lead to coordination among researchers that are involved in pain research. So you've already heard about the IBBS Center for Sensory Biology, which is sponsoring today's event. The Blaustein Pain Research Center is a center at Hopkins that both holds weekly seminar series on topics ranging from the basic science of pain to biobehavioral aspects of pain to clinical aspects of treating pain. There's the Basic Sciences Institute Pain Working Group that Paul Fuchs referred to earlier that has allowed a group of like-minded individuals to assemble to try and learn from one another and essentially bounce ideas off of one another and see where collaborations might be effectively created. And then finally, there's the Department of Neurosurgery has just started the Neurosurgery Pain Research Institute, which is emphasized not only on understanding pain better, but on developing better neurosurgical practices to treat pain. And so I'll just close with that, and I'll be happy to answer any questions you might have.